Wonderful. Thank you. Thank you very much for the introduction. Can you hear me well? Can you see the screen correctly, just to check?
Yes, yes.
All right, fantastic. Yeah, I mean, first of all, thank you very much for the invitation, and also really thank you for organizing this session. I really enjoyed the last edition. I'm happy to be back here, to be honest. Unfortunately, I missed most of the talks because I had teaching on Wednesday afternoon for the entire photothermal webinar. But I promise I watched almost everything I could find on YouTube. So I hope you'll upload the rest of the videos soon. In any case, I'm going to talk a little bit about phototransient holography. So within my research group, we focus on many different aspects that are kind of related: bioimaging, nanosizing, spectroscopy, holography, and also ultrafast. So, I mean, I worked with Philip during my PhD, and actually I didn't work on iSCAD. I worked on femtosecond spectroscopy back then. And somehow, I don't know, during my time with Nick in Spain, that came partially back. So this is what got us into this phototransient holography business. Just up front, I wanted to kind of thank the people that really made this whole technique possible. And that's, I have to say, mainly the group from Milan, with Julie Chirullo, and let me see, I have the pointer here, with Giulio, with Martin, and with Franco, who were kind of the people I contacted back then when I had the idea, and I was like, come on, we need to try, I need a laser, I need some help. And I contacted Giulio, and we started working on this together. And of course, also many thanks to Nick and to Alex, who used to be a PhD student in Nick's group, who kind of gave the first ideas towards what we then started to work on in the long run. Just because this is the photothermal webinar, I thought maybe I share like 1, 2, 3 cartoon slides with you to kind of motivate why we are trying to do phototransient microscopy and how that differs from kind of photothermal microscopy. In a way, it's very similar, and there are some minor differences. So in this cartoon, what you can see is you can see a cell, and I just, back then, I don't know, this is a very old cartoon, I drew a few viruses there, and I put gold nanoparticles on the viruses. And the idea was to, okay, we can now like optically heat the gold nanoparticles. If we would be able to do that, and we would be able to photothermally detect where the virus is. That was the idea. And then as I started thinking more and more about this, I realized, okay, the longer we wait, the more we will cook maybe the entire cell or heat the cell, depending on the way we illuminate. So there was always this kind of question of how does time impact the measurement? And very similarly, if you come from a more femtosecond world where I kind of grew up during my PhD, we have very similar problems. So for example, if you were to locally heat molecular bonds, here in this case, maybe this carbonyl stretching mode, you would very, very rapidly take this energy that is transiently deposited in kind of a non-equilibrium state, and you would thermalize first over the molecules, maybe within picoseconds, and ultimately you would start transferring to your surrounding and heat the entire sample, while kind of losing your local specificity. So my conclusion back then, and I think still, is that timing is really, really everything. Yeah? So depending on your time scales, be it nanoseconds, microseconds, or femtoseconds, you will have a very different kind of signal from your optical excitation. And if you think about photothermal microscopy, I think we are mainly talking about the nanosecond range, because this is something you can kind of access, or you access it by megahertz modulation with a lock-in amplifier. And that's perfectly fine. It works, I think, fantastically well. And most of the implementations are kind of confocal implementations, which kind of capitalize on the fact that you're transferring some of your heat to the surrounding, which then ultimately gives you the photoresponse that you would like to see. So what I really like is cameras. And unfortunately, cameras are not as fast as lock-in amplifiers. And they, if you're really lucky, they work somewhere in the microsecond range. So you can go to kilohertz frame rates. And if you remember everything I just told you, that is problematic, because as you can see here, if you're trying to do a local photothermal kind of process, no matter what you heat, you're always going to end up at the place where you've already heated your entire sample, even though you wanted to see maybe nanoscale changes, nanoscale structure, you name it. So that is kind of a problem. Nevertheless, I think cameras are fantastic. And in the past, I used to always talk about what you can do with it, but I mean, we recently started to really use cameras. So I thought I'd just show you a video of what you can do with cameras. And these are the kind of videos we can like readily record these days. So these are kind of quantitative phase images of live cells. We record over hours. We can see all of these changes in real time. You can see very funny things like these cells up here dividing into 3 cells for whatever reason. You can get a massive amount of information. And on top of that, I believe it is very straightforward to implement a kind of camera-based microscope as compared to a confocal microscope, which is very alignment sensitive and also very sensitive to things like chromatic aberrations. Once it works, it's fantastic, but it might not necessarily be for everyone. And of course, what I show you here, you can really take this to the extreme and you can record gigantic images and you have a wealth of information that is very difficult to access in a kind of confocal imaging way. So this is where phototransient holography came from. And our idea was to say, okay, let's find a way to combine the camera and move it kind of to the fastest possible timescales. So basically saying, how can we record transient events with very slow readout while simultaneously ensuring, let's say, sufficiently high signal quality so we could actually obtain information. Given that this kind of technique combines 2 aspects, one is the kind of, let's say, the femtosecond world, which we call phototransient or non-steady state, photothermal microscopy, and holography. What I'd like to do is I'd like to introduce you briefly to the holography aspects to make sure we're all on the same page. And then what I'll do is I'll put the phototransient aspects on top of it so that we can walk through it together. So just to kind of introduce what a holographic experiment or holographic microscope would be, I decided to just work with this slide. So it's if you like, it's the simplest form of a holographic microscope. So all you have is you have a microscope objective here, any objective you like. You have a lens here, again, basically any lens you like. And then you have a detector here, a camera in our case, which records an image of whatever is on top of your microscope. And then, as you know, from microscopy, there are normally 2 ways to look at your sample. You can either transmit light through the sample from the top, in which case you kind of detect scattered light and illumination light, or you could illuminate from the bottom. And again, you would detect scattered light and kind of back reflected illumination light. So there are some differences here, of course. But in the grand scheme of things, we can say, okay, whatever we detect from our microscope, we call it the signal electric field. And then all that holography does, it sends a reference wave onto your detector. With the idea being that you're able to ensure phase-sensitive detection. So mathematically, you can think about it as shown here. You'll record, of course, an intensity, your image, and this image will be composed of the square of this signal field, the square of the reference field, and a kind of interference term between the signal and the reference, where ultimately you have a cosine in here, which gives you your phase. And now if you look very closely, you see this kind of maybe strange term up here, which is the phase of y. And what I mean with this is that if you think about your camera and you say, okay, this direction is my y plane, you kind of have a phase gradient because of this angle that I introduced here between the reference k vector and the signal k vector. So what that means is you get a y-dependent phase ramp. So if you think Fourier transformations, it should be clear that you can extract this term here with Fourier filtering purely based on this modulation, which is kind of a cosine modulation. Depending on what you want to do, if you think iSCAD or you think off-axis holography, you think inline holography, it's all the same. So for example, in inline holography or iSCAD, all you do is You would move your reference into the center of this E signal. As a matter of fact, you would use the back reflected light. Now you have iSCAD inline holography. You could put a dark field mask here, then interfere. You have a different modality. You can do what we are doing, which is just transmit the light, use the reference. You have off-axis holography. The information content is slightly different, but the concept is kind of identical. Why I really like it is because you can measure things really, really well if you go for the off-axis way. So you can get very quantitative signals. I'll show you 1-2 examples in a minute. And as a bonus, which I think makes it really distinct from more the inline approaches, is you can do a lot of kind of computational, let's call them tricks, where you do digital post-processing to, for example, record at one image plane here and then computationally propagate your image to generate a kind of a 3D image stack under certain experimental conditions. I'm going to go into detail later. Just to kind of take this digital post-processing maybe to the extreme, one thing we did is we built a microscope that we called a kind of K-space microscope, which in a nutshell is very similar to what I just showed you. But if you look closely, what you see is we have 2 lenses here. And then what it means is that on our camera, rather than actually detecting an image of the sample, like something we are used to, we record an image of the back focal plane of the microscope objective, which is, I think, a very non-intuitive way of looking at a sample. And then again, we use off-axis holography to interfere. And if you look closely what we record, I think it looks kind of terrible. Like, at least for me, there's no information whatsoever, apart from the fact that there's something round, which might be the back focal plane. However, if you then start Fourier processing your data, if you remember this equation, what you'll see is that in the Fourier transformation, you actually see something that might look like particles. And in this case, we have particles that are freely floating in a kind of in a container. And then you can start playing more, and you can say, okay, now I'm going to propagate my sample. I would like to look at different Z planes in the image, and so on and so forth. So you can really go from something that looks like a kind of failed imaging experiment and reconstruct a 3D volume of nanoparticles with a few computational kind of steps. And these things work really well. So we applied this, for example, to quantitatively size extracellular vesicles. This is an example. We have an enormous dynamic range. You can see we have very large extracellular vesicles here. And at the same time, we can detect tiny extracellular vesicles. So if you are like in the sizing world, these are kind of 4 orders of magnitude difference in scattering intensity. So this is an enormous difference in signal. And we are able to cover all of this with a simple microscope like the one I just showed you. And then if you look a little bit closer, and this is something I really liked, if you compare our sizing efficiency, meaning if we determine how large the individual vesicles are from their scattering amplitude, we obtained results that very nicely align with what people have commonly seen these samples based on kind of TEM measurements, which I always like to think that it's a great thing to be able what a TM can do with a very cheap kind of platform. Maybe to kind of take this kind of 3D concept to a next level, I just wanted to show you this. So this is a similar experiment. And the idea here is that we have really a volume of particles. The particles are freely floating. And what I show you on the bottom are 2 different views of the same sample. So on the left is what you would see with kind of a conventional microscope. So you can see there are particles freely diffusing. Sometimes they're in focus, sometimes they're not in focus. There are a lot of speckle. And on the right, you can see the same image, or not the same image, but the same data. But here what we did is we propagated over the entire volume that we are illuminating. And then we just kind of did a maximum intensity projection. So this is not the cleanest of images that you can get, but I hope that it gets the message across. So whereas on the left, your particles tend to disappear on and off. On the right, you are in focus all the time, and you can really track for very, very extended periods of time. And that is extremely useful if you think about sizing, because, you know, if you want to know the size of a particle, you always have this kind of dilemma if you work in kind of scattering or holography, and that is that you you have a reference, and then you trust that the material you're measuring is actually the same material that you think it is. So if that changes, and in biological samples that's often the case, then you will get the size wrong because the scattering amplitude changes. So what we can do now is we can use the scattering amplitude to determine the signal of the particles. And at the same time, we can use the diffusion coefficient to determine the size of the particle, aka the hydrodynamic radius. And then we're able to kind of decompose samples which contain different materials and different sizes into their respective contributions. So if you look here, if you look closer, for example, you would see that the tiny gold nanoparticles that you see on the bottom left, the 40 nanometer particles, have a comparable scattering amplitude as the 140 nanometer silicon dioxide spheres. So if you would measure them together purely based on, let's say, a standard scattering experiment, let's say iSCAT, you would not be able to tell the difference. Whereas if you then at the same time measure the hydrodynamic radius, you can clearly see that they're very different materials. That one is almost a factor of 5 almost larger than the other. And what really changes is the refractive index. And the lines just show you that what we measure also really nicely agrees with kind of theoretical predictions of the size-dependent kind of scattering amplitudes that we would expect. And this works for both heterogeneous and homogeneous samples. So here on the top right, you see a heterogeneous sample that we measured, composed of 40 and 60 nanometer gold spheres. And you can see that within 3 sigma, we are, I think, pretty decent when it comes to distinguishing these two contributions. Okay, so this is kind of, I think, the holography aspect of the talk. And now I'm going to move a bit into the phototransient part. So just coming back to the cartoon, and I think I'm going to show it once or twice more. The idea is very simple. You have a complex sample. It doesn't have to be a cell. It can be anything. And you are interested in local changes or nanostructures or micron scale information. And what you'd like to do is you'd like to heat, and then you'd like to detect your structure or your information based on this kind of light-induced heating. And it works really well with photothermal microscopy. And I think one of the key ingredients of photothermal microscopy is very high sensitivity. And that's, in a matter of fact, why a lock-in amplifier is the absolute ideal tool, I think, to ensure that you are really at the shot noise limit and you get the information you want. So for the phototransient implementation, we were kind of wondering, how can we achieve a similar kind of readout? So how can we take this concept of like time domain lock-in amplifier and translate it into kind of an all optical, what we call an all optical lock-in amplifier? So it might be a bit tricky, but if you think about here, you're relying on time, energy, uncertainty. So this is why you can extract information. And here we don't have that luxury, but what we can do is we can rely on position, momentum, uncertainty. It might sound abstract, but it is basically at the essence of holography. And that is what we did. So this slide, I think, is a bit tricky. So I'll try to walk you through the key components, and then I hope that everything will become clear. So you can think about it as the holographic microscope that I showed you before. They're just more beams. And to start, I'd like you to ignore this excitation beam here, the yellow guy. This is just to basically induce the signal, the phototransient signal. And we just focus on the imaging for now, which is the blue light. So there are two things here. We have what we call the probe. The probe illuminates our sample. In this case, it's in transmission. And then here we generate the reference wave. The reference wave is the same reference wave as I showed you earlier when I kind of briefly outlined a standard off-axis holographic microscope. And now the only difference is really that we use a 2D grating and we generate not one reference wave, but we generate two reference waves. And we then propagate them onto the camera where they interfere, as in the example I gave you before. And now the difference is that we have two different kind of, if you like, K vectors or angle. So these reference waves interfere at different angles with our signal, and therefore they will modulate the signal in different frequencies, in different K places in the Fourier transformation. To make this a little bit clearer, maybe you can think about it like this. Imagine there's an image, in this case, nanoparticles. Your one reference comes from, let's say, one diagonal, the other from the other diagonal. And what you get is you will get interference fringes in one dimension if you're coming from this side, and in the other dimension if you're coming, let's say, from this direction. And you can see this if you zoom into these nanoparticles, you're going to see some checkerboard pattern. It might look a bit strange, but then if you Fourier transform, you can see that you have these interference terms which nicely separate into kind of two contributions. And to make this phototransient, what we do is we now modulate the individual reference waves in a way that we switch on one of the two reference waves when we also switch on the pump. So when we illuminate the sample and photoexcite the sample, and then we switch it off and we switch on the other reference wave when the pump is off. So we kind of have an on reference and an off reference. While we're doing this, we're continuously illuminating the sample with our signal or probe, if you like, and then we just integrate. And then we extract the information from the holograms. And if you look at it, this works, I think, really, really well. So what you see on the left, I think these are 60 or 80 nanometer gold nanoparticles. I'm sorry that I forgot. I'm not sure. I think it's 80. And what we did now is we continuously imaged, or wide field imaged these particles, extracted our holograms, and simply looked at the pump on and the pump off contribution in the hologram. And we asked ourselves, okay, how is the signal going to change if we move a pump beam across this kind of image? And the idea is very simple. If your probe arrives before you photoexcited the sample, you will not see any signal due to the pump. And if the probe arrives after you illuminated your sample temporarily, you will see a signal change. And this is what we see here. So before time zero, both the pump on and the pump off channel show identical signals. And then as we move over time zero, we see this photoinduced signal here, the plasmon resonance in this case of these nanoparticles changes. And we see that in the pump on channel, where the reference illuminates while the pump illuminates, we see a signal change. And in the other channel, we don't. And this is on a single nanoparticle level, of course, in a wide field geometry. And just to kind of ensure that this is not some kind of funny artifact, but really works, we then did kind of systematic comparisons between nanoparticles that we could photoexcite, in this case gold, with a plasmon resonance, and we then compared this to dielectric nanoparticles which did not have an electronic resonance. In this case, at 400 nanometer, where we excited. And you can see that on the top, we can observe both nanoparticles, but on the bottom, in this kind of differential scattering signal, so the photo-induced change in the scattering, we only see a change in the gold. And if you look at this quantitatively for many, many, many nanoparticles, you can kind of recover what I just showed you on a single particle level. So these are just many trajectories, whereas the dielectric spheres do not show a change. And then you can play all the tricks I showed you before. So for example, you can go out of focus, which is then, this is your amplitude, this is your kind of phase contribution from your hologram. You can then computationally refocus this. So you have a nice, hardware refocus this. So you move your microscope focus, or you can take this hologram and simply use the computer to refocus. And you see, you kind of get the same result. So on the right is the hardware focus. On the left is the software focus. And this works both for the standard scattering, as well as for the phototransient imaging. So here you just see a wide field out of focus. It looks very random. You see nothing here. You see kind of a speckle-like pattern that lights up as we photoexcite and then disappears. We can computationally refocus and we recover all the nanoparticles. And again, we are back to where we were 2 slides ago, but we're not in focus. And you can really take this to the extreme. So here we have free-floating nanoparticles. We excite the free-floating nanoparticles. We can look at the trajectories of all these individual nanoparticles that are floating somewhere here while changing the pump probe time delay. And then we can measure kind of transit responses of the individual nanoparticles in free flow, in 3D, you name it. So from this experiment, we kind of concluded that this experiment here should work. If we locally heat something that is resonant, we will be able to detect it. If it is not resonant, we will not detect it. So in my cartoon, we should be able to find the gold nanoparticle that has labeled the virus that is on top of the dielectric cells. I think I have something like 5 minutes left. Is that correct?
Yes.
All right, I'll try my best. Yeah. So with this in mind, we also realized something else, and that is that I've talked a lot about 3D localization. Holography can do everything for you, but this is unfortunately only true if your sample is kind of sparse. It's the same as for us, right? If somebody is behind a wall, we cannot see that person, even though we are somewhat able to see in 3D. The same is true for microscopy. If your sample is very dense, you cannot see objects that are behind other objects within the depth of focus of your optics. So kind of the dilemma is here that we can do really cool probe kind of microscopy, holography, but if our sample is three-dimensional, we will recover time-dependent complex refractive index changes, but unfortunately only in 2D. But what we actually really wanted was to have this information in full 3D, irrespective of what the sample looks like. And this is work I did together with Martin. And I think on this picture, he's smiling during this project. I think he was very often not smiling, but we managed to kind of conclude it. And I go through it very, very fast because we have very little time. So on the left, I just wanted to show you that this is not the most straightforward experiment. It's because we're using highly broadband femtosecond or like pulses with a transform limit around 5 femtosecond. We try to use high-NA objectives. We try to use do optical diffraction tomography with these pulses. There are a lot of experimental issues, let's say, things like wavefront matching or chirp. It is just tricky. So in a nutshell, what we do is we illuminate our sample from different directions, and then we record holograms. So very, very briefly, we record a massive set of holograms, then we extract the interference term as before. We extract phase and amplitude information in real space, and then we go through several computational steps to ultimately end up in the kind of 3D representations of the refractive index of the sample. So in this case, cells. And because we're very broadband, we can then kind of spectrally decompose this information. So here you just see X, Y, and ZY projections of these cells at different wavelengths. And we were asking ourselves if the kind of, let's say, the average refractive index of these samples has any sensible dependence on wavelengths. And what we kind of saw is that, yes, it does. So as we move from blue to red, we see a change in refractive index. And these are kind of both off-resonant materials, let's say all the kind of cellular components in water. So we kind of see only a relative refractive index change with respect to water, is what we would expect. To ensure that there are no artifacts, we then went 5 steps further and basically build a microscope where we can do narrowband optical tomography, optical diffraction tomography, and at the same time, broadband optical diffraction tomography with Fourier transform spectroscopy. And we ensured that the spectral information we retrieve matches. So here in the upper case, all the artifacts you would expect are reduced because the temporal coherence length of your pulses is very short. I mean, very broad still for optical diffraction tomography, but very short compared to this kind of spectrum. Nevertheless, irrespective of what we did, we kind of saw, I think, a very nice match between the kind of narrowband approach, the broadband approach, and a pure kind of spectrum of the laser pulse taken with a spectrometer. And then I will skip this slide, and you can then apply all of this to really retrieve 3D images of cells. And in this case, we haven't done the photothermal yet, but we are at the point where we're able to localize gold nanoparticles based on their kind of 3D complex refractive index response. So we're able to distinguish, let's say, cellular materials from nanoparticles, from kind of artifacts in the image which are due to processing. Well, I guess with this, I would say timing is everything because I have to come to an end. I'm sorry that I had to rush the last few slides. I would like to spend a little bit more time. Before I stop, I would really like to thank all the people involved. This was, I mean, Julio was extremely helpful with implementing all of these phototransient images, all of this stuff. There's the old group where I worked in Spain, which was contributing on the original holography, especially Unai. There's Jaime and Romain, who are involved in all of this work in one way or another. And there's now also my new group. We are missing one group member here. She just joined it. So the picture is missing Yinke. And I just wanted to announce that we're still looking for people to join our team. So we are building up a phototransit imaging system for biological applications. And if you're interested in biology, medicine, or physics, or chemistry, get in touch with me. Thank you very much.
Thank you, Matz. It was very exciting talk and very beautiful results. We are open for questions. So please feel free to ask. I think Michelle has a question. Yes.
Yeah, thanks a lot, Matt, for a great talk and exciting results and possibilities. I was wondering, just a question, when you showed this transient on the picosecond time scale, so this pump probe spectroscopy, so I expected to see these coherent oscillations of the particle in the case of gold and metal particles that absorb a lot. So we don't see those. So what's happening there? Is that just because there is not enough signal to noise ratio, or what is the reason?
It's a good question. I had my feeling was that I think we are at the time scales we are at. I think we're a little bit far away even from that. Like if you think about like I don't remember the original works, but I think the time scales are much longer than the 40.
Yeah, typically it's some tens of picoseconds, depending on the size of the particle, but so some tens of picoseconds.
Yeah, but, I think there are two answers to this. I think one is that it might be difficult to see minor oscillations, which might be things like this here. Yeah. That is one. The other one is that this modality really relies, at least for these particles, on the kind of hot electron contribution where we get a massive signal change due to the plasmon resonance shift. So
also the plasmon enhances these oscillations are visible because of the plasmon.
Yes,
without plasmon you couldn't see them.
Yes, I agree, but I have the feeling that maybe on the photothermal you're more likely to pick them up. So our signal drops dramatically, as you can see, right? The overheating we have is very small. Our sensitivity for the pure heating effect is not so small. I think that might be one answer. The other
so
answer
what we're seeing is the electronic response, right?
The electronic response is gigantic for us.
Picosecond.
Yes, the electronic response is gigantic for us.
So this signal should be in the tail that you should integrate long. Okay,
yes,
okay, I see.
Yeah, and I mean the other thing might just be that we need to maybe extend the time delay to like, I don't know, 200 picoseconds to actually
the excitation pulse is very short, right? I mean, that's what we see here.
Yeah, these were like 100 femtosecond pulses.
Yeah. Okay, of course, the dispersion of the objective and
so
lengthening. But,
this is one of the things I actually forgot to mention. One thing I really like about these kind of non-confocal approaches is that you can be kind of ignorant about your excitation, because we can very often go free space onto the sample and then just detect with the objective, which means you can use infrared or deep UV or very, very short pulses, because once you have excited, you don't really care anymore what happens to your excitation beam. So that's kind of it's a nice advantage over photothermal. There are many drawbacks, but that's something I really like.
Thank you very much.
Yeah, Randy has a question.
Yeah, thanks for that great talk, Matz.
Thank you.
So this image at the end, like your last or second to last slide, is this of the cell is really provocative. So great. You can tell the difference based on the complex refractive index between nanoparticles and cellular material. Is there a path to telling the differences between, for example, like a really dense organelle like a nucleolus, or maybe a liquid biocondensate from a less dense organelle like ER? Is this something you're thinking about? Is this feasible?
I mean, these things are, well, the straightforward answer is yes, but I mean, you probably know maybe better than me even that this is really tricky business, right? I mean, I definitely
do not know better than you that it's a tricky business, but I believe it's tricky.
Yeah, so the issue is a little bit that all of this is nano scale. So we measure refractive indexes, but as soon as you go into the mixtures, you get kind of refractive index mixtures, right? So imagine you have a nano water bubble and you fill it halfway with protein. Your refractive index will be somewhere between protein and water. So yes, we can distinguish if let's say within a condensate, there's more or less water. I think that would be a really cool application that we thought about at some point. But it might be very tricky to distinguish this from some lipid droplets. So maybe in combination with a kind of fluorescence marker that tells you, okay, here's a condensate that I'm interested in, you should be able to get things out like what the condensate is composed of. If you narrow it down, I think that should be feasible. Absolutely.
Yeah, I mean, particularly with the time resolution that you have, label-free ways of getting at these condensants, I think would be very, very impactful. So I'm glad you're thinking about it.
I mean, we were thinking about actually to kind of maybe use also a diffusion processes, not just look at the refractive index, but maybe try to see if you can get into things like local fluctuations, these kind of things. It's to be seen.
Yeah, exciting.
Okay, is there any further question? Maybe in meantime, I ask a very quick small question is that how is the sense if you think about the sensitivity? Yeah. So even if you think about, let's say, gold nanoparticle, how small particles can it be detected? And what would be also in terms of spatial resolution?
I mean, I think the standard answer is always short noise, I guess. So I mean, within very reasonable integration times, we went down to 20 nanometer gold. We didn't try to push it further. We are now building up a setup where I think we should be able to go to roughly 10 to the -5, maybe a little bit lower, maybe 5 times 10 to the -6. Within a second over a relatively large field of view of maybe 150 by 150 micron. Going further becomes, I think, very painful because, I mean, then you integrate for minutes. I think for these super high resolution, amazing things that you are an expert in, I think for that, the photothermal one particle confocal approach with a lock-in amplifier, there's just no way to beat it because you get all the photons you want, you get the sensitivity you need, and you don't really care about a gigantic field of view in this application. So I think that's probably the way to put it.
Yeah, thanks. I think Gordai has a question.
Yeah, hi Matz. Yeah, thank you very much for the presentation. So I was wondering about the 3D tracking of gold nanoparticles. I think you mentioned that there is also a high-speed option. So, and I'm wondering, so is it recording high speed, or you also do this in real time, all the analysis, or probably this is post-processing, or?
Yes, I mean, high speed, we can go fast as your camera can go, but yeah, if you have any idea how to speed up the analysis, that would be fantastic. It is,
yeah, because it's a lot of Fourier transform, yeah,
it is,
for every frame.
Yeah, I mean, we're looking into all the kind of deep learning to accelerate these things, of course, because some of these things are so obvious that you wonder why nobody has done it yet. And I mean, there are people working on it. So I hope that will speed up the process quite a bit. We will see. But yes, the post-processing is certainly, it's not forever, but let's say 1 second of data is like maybe 10, 20 seconds of analysis. It's not terrible unless you want to record the entire day. But it would, of course, be nice to streamline it and make it fast.
Okay, yeah, clear, yeah, thanks.
Yeah, Michel has a question.
Just a question about the 3D imaging. Also, it's very nice that your holography method allows you to have the Z to image in depth. How does this compare? Can you make a sort of short comparison between the point spread function engineering that W. Mern and others have been pushing forward and holography? So what would be the pros and cons?
I mean, it depending on which aspects you mentioned. So the point spread function engineering of Mörner, I think, is mainly, I would put it into something I would use with fluorescent light mainly, because I just don't see need to if you're scattering, you can use interference, you can use computational tools. So I don't really see a need for point spread function engineering, which requires actually very sparse samples, as a matter of fact, because you're really spreading your information all over the place. If you're going for large set ranges, that's, I think, the very simple answer. The advantage of point spread function engineering is, as far as I'm concerned, that if you're dealing with low photons levels that you cannot amplify by heterodyne detection, like in holography, then you can localize your information on very few camera pixels and then access a large Z range. Whereas if you would be just projecting the light onto a detector, you would be buried in the darkness of the detector. If you can heterodyne, you don't care. You just amplify and you can detect one photon. It doesn't matter how far you are out of focus. That's my view. I think it's fantastic for incoherent light.
Yeah, thanks a lot. Very clear.